All right, environmental cultures will be our first activity. And one of the reasons I always start off with this one is because it doesn't require really much in the way of lab safety. As long as you don't trip and fall, I guess you'll be fine, right? So in this particular case, I'll show you this super easy, where we're going to take a sample from the environment, and we need to know what it means to culture something from the environment. So let's take a look at this word culture. Okay, culture is a term that both medical doctors will use, like to culture, say, a patient sample, but what are they culturing? Or in lab here, what are we culturing when we culture a sample? It is a collection of growing microorganisms. We could probably assign more words to types of cultures, but this is a basic working definition. And you can grow microorganisms in various tubes, and here we have a Petri plate that we'll be using for our environmental cultures. All right, so let's see how we're going to be setting those up. What I'm really trying to do in this experiment is prove to you guys that all around in our environment there are microorganisms. They're everywhere. Anywhere you touch, right? You have microorganisms on you, but then anything else you touch, you may be picking them up from your environment. And so we're going to follow these steps to taking a sample from the environment and then seeing, what's lurking, that's microscopic. What could be there, but you can't see it, right? It's micro. And yet we're going to grow up these organisms to visible numbers, all right? So let's see. Let's follow the steps, okay? So it says here, label the bottom of a Petri plate, all right? So I have my Petri plate here, but I'd like to kind of do a close-up of this. And so I'm going to put this on the document camera. So we'll switch over to the document camera. And here's our plate coming into focus. All right. Now, back on our instructions a minute ago, or a second ago, it said label the bottom of the Petri plate. A Petri plate has two halves to it. So let's look at those two halves a little more closely. First we have the empty lid, and you can see some water droplets on the underside of the empty lid. Don't worry about those water droplets. That is actually sterile water. It will not interfere with our experiment, but this is the empty lid. Here we have the bottom of the Petri dish, and this bottom part of the plate has a thin layer of agar, which is a jelly-like substance that has nutrition in it for microorganisms to grow. Now, as long as this plate remains closed, it's considered sterile. Sterile means that this plate was made carefully enough with the right procedures for sterilization to make sure that no microorganisms are in it right now. Now, I took a risk when I opened the plate to the air, and that's something that maybe we can test as well. Some of you might have heard that germs can float through the air, right? We're worried about that with COVID-19, right? What if someone who's infected is in a room with like closed air where, viruses could be floating in that air, and then someone else could inhale them. And in this case, we're going to be opening these plates. We're not going to be able to grow viruses, but we are going to be able to grow some bacteria. And speaking of the air, let's test the air. All right. So I've already opened the plate a little bit anyway. So I'm going to just go ahead and label this. Oh, and by the way, it said label the bottom of the plate. So the bottom is the part with the food that I'm holding up. All right, that's where we're going to be labeling. So I'll put D for Derek and B for Boyer, my initials, and then I'll go ahead and put air sample air sample 20 minutes. Okay. Now, before I open this up and expose it to the air for 20 minutes, let's think about a couple things. Why did we label the bottom of the plate specifically and not the lid? Because most of the time you label the top of something. And so one reason that we're going to see is that once we take our sample, we're going to be incubating these plates with the lid down and the bottom of the plate up. And the reason is because of the condensation that I was showing you. See how here there are water droplets. Those water droplets, they'll build up more and more if we incubate the plate this way. Because when you put the plate into this gray box that I'll be showing you, the incubators, I'll put them on camera in a bit. What happens is the plate heats up to a nice toasty 37 degrees Celsius, which is actually human body temperature, coincidentally. And what happens is the agar that's on here, the jelly-like substance, has a lot of moisture, and water will come up out of that, and it will collect on the lid here. And the underside of the lid will get more and more water droplets that will start to get heavier, and then they will drip, drop, and rain down back onto the surface of the Petri dish. And then we won't really have individual bacteria growing in colonies on the plate. It'll just kind of be a swimming pool of bacteria mixed together, and that is not what we want. So we do not want to incubate the plates right side up. We can prevent this dynamic by simply flipping the plate over. And if you're wondering how it is that flipping the plate over makes a difference, it has to do with the fact that the moisture source is now on the top. It still has moisture, but the way that the moisture will escape and not build up is that if you look really closely, it's actually a little hard to see. I'm going to go ahead and turn the light on this, and maybe it'll give us a chance to see a little more readily. I'll flip it over for a second so you can see it from this angle. Okay, right up here, there are feet. There's a little line there and a little line there, one there and one over here. They're little notches on the underside of the Petri plate lid, and what it does is it keeps these Petri plates from sandwiching to an airtight seal. They do not seal shut. This plate actually has room for gas exchange, and so when we flip the plate over like this, gas will be able to escape, water will not build up underneath the lid, and it's nice that we can see our label facing up as well. And if there's one more thing that we can keep in mind here, it's that what if we had an accident? What if you had a stack of plates and they fell off the counter or something like that? If these plates come apart and you had an accident, if you have the label on the bottom, that's where the bacteria are growing on the actual food source. The empty lid is less concerning. You can label both sides if you like, but at a minimum, we always label on the bottom because of this fact. That's one of the safety concerns that goes into this. Okay, so now you've seen this a little more close up, and we'll go back to our instructions. So now what we're going to do is wet a swab. So we need to have some sterile saline, and give me just a moment. I'm going to get some lab supplies and put us on a close-up of what's going on. Okay, so step 2 is to wet the swab now that we've labeled the bottom of our plate. And I have a container here of sterile saline, and this is basically a water-containing solution with just a touch of salt that puts the right amount of osmolarity osmotic pressure. We'll talk about that later in the course, but for now you can see that we do need a little bit of water. We want to wet the swab because when we collect our sample, we want that, that sample to pick up nicely off of whatever it is that we collect. So here I'm getting a sterile swab. Now when I take this out, I want to do a careful job to not touch the sterile swab to something that's not part of my experiment. Now, I just realized that this was my air sample. All right, I'm going to go ahead and set this aside. I think I'll put this over here, and we'll let this stay open for 20 minutes, okay? And so that's the 20-minute air sample, and I just looked at the clock, and I'll have to collect that in just a little bit. Now, I have another plate that I'm going to use to sample something else. So I wet the swab. I'll carefully take off the lid, and I'll go in and wet the swab, and I don't want it to be dripping wet, so I'm going to kind of squeegee it off a little bit. And then now I need to sample something. So I'm going to sample, what shall I sample? How about my cell phone? So undoubtedly my cell phone has body oils on it and bacteria, and there are all kinds of little cracks and crevices and things where my cell phone has picked up bacteria and other germs and such. So I'm going to go ahead and take this wet swab and just wipe along here. And I'm going to wipe kind of hard because the wet swab isn't going to mix that well with body oils. All right, oil and water don't really mix very well. So I'm getting kind of just extra thorough to go to different surfaces and just see, these are all places that my face or my fingers could touch, and we'll see which kinds of bacteria will be picked up from the cell phone. All right, so we're not being very precise about the technique of collecting the sample in terms of how many swipes or swabs, swabbings, but what I'm going to do is turn my plate over, and let's start with it lid face down, and I'll carefully then leave the lid down there and take the open auger, and we'll wipe it around, rotating this around. There's really no wrong way to do this unless you just press so hard. It doesn't take much muscle to move the bacteria around and spread them around on the plate, but sometimes students will press so hard that they split into the jelly-like auger. So I'm using a light touch, but I'm being very thorough to make sure that I get all my sample off of there, okay? And so now I go ahead and I take my used swab and I put it back into the container that it came from, all right? And then I need to relabel this. So this was my cell phone, and I'll put my initials there. Also, I would like to try a sample, just another sample for good measure. Let's try, how about a shoe? How about my left shoe? Why not? So I'll go ahead and sample the bottom of the shoe. All right. I would expect that we're going to get a very different sample from the cell phone, which I'll set aside over here, and then the bottom of a shoe. So once again, I'm going to take a swab, carefully take it out, and then I'll sample the bottom of my shoe. So I wet the swab, okay, and then give me a moment here. Okay, I just swiped several times on the bottom of my shoe, and I picked up a lot of dirt. It's visibly dirty. No big surprise there. It is a shoe. So I'm going to go ahead and spread this around. Notice how I'm keeping my fingers away from the sides. This experiment is, the way that it's being conducted is to avoid getting anything else into the experiment, right? I'm just trying to test the bottom of my shoe, not, bacteria from my hands. So I'm being very careful to handle the edges without getting bacteria from my hands into the sample. Now I'm going to go ahead and put the dirty swab back into this, and then these tubes are going to be put into a sterilization machine called an autoclave, and so those will get cleaned out. This should still be sterile water so long as nobody did anything like dipping, non-sterile items in here, and so usually I pass this around to other students. All right, and the very last thing we're going to do is we need to incubate. Now remember, we're going to be putting them into the incubator upside down, so we'll stack these plates up in stacks like this. All right, so I'm going to go ahead and take these to the incubator now, so I need to press pause for a moment. Okay, here we are in front of the laboratory incubator. We have several of these in our lab. This one is turned on right now and set to 37 degrees Celsius, which is approximately 98.6 degrees Fahrenheit. That's human body temperature. We're going to be growing these organisms at a very toasty temperature. And so what we're doing is taking the stack of plates, and we open up the insulating jacket, and there's an inner door, an inner glass door that will help to seal in moisture. In fact, we can put our stack of plates here, but we can also look down in here and see that besides our plates that we're incubating upside down, there is a source of water, just a little bit of water in there, and that can evaporate over time and keep it nice and humid inside the incubator. The bacteria like it warm, they like it with a high moisture content, and at 98.6 they will grow at their best rate, so that 2 days from now we will have some nice colonies growing on there that we'll have a chance to make some observations on. Okay, now that we have our environmental cultures in the incubator, this one still has to be out for a little bit of time, the 20-minute air sample. But we have all the other ones in the incubator. I'll put that one in just a little bit. We can move on to our next topics. So environmental cultures, day 2. When I come back and we look at this, we'll be trying to observe what the colony types are like. So I'll skip over this for now, and we'll come back. We probably should take time, though, to answer these key questions here. So why do we incubate plates upside down? It is because it prevents condensation buildup. Condensation is the buildup of water underneath the lid, and we do not want that. So we incubate with the lid down so that that can't happen. Also, another good reason to incubate plates upside down, oh, I didn't talk about this. When you stack the plates actually stack better with the lid down because the lid is larger. To give you a bit of an example, if I have a stack of plates and the lid is face down, it's easier to grab the very bottom lid on a stack of 3 or 4 or 5 plates than if they're right side up, because the lid is larger, like an umbrella, and you go to grab it, there's a chance that as you grab the stack, you could end up lifting the lid off and leaving the smaller bottom down on the counter, if you're following what I'm saying. It's another thing. It increases the plate handling. Dexterity issues are solved by having the plates incubated upside down. The label also faces up, right? So we have labels face up. We have stacks of plates are easier to handle. And you could probably argue that the label facing up could be on this, the answer to this next question, but there's another concern here. The second question says, why do we label the agar side of the plate? And if you recall, if there was an accident and the lid got separated from the bottom, then the bottom has the agar, right? The bottom of this plate is where there are going to be bacteria actually growing, and the lid is empty, and so it's good that we have the label on that side, right? On the side that has the bacteria growing. The label is present on the part of the plate where growth is present. And then this last question will be partially answered next time, but it says here, how do we know it is bacteria, not fungi? Actually, maybe the wording here could be that the bacteria are likely what we're going to be growing out of our samples, more so than fungi. And this has to do with how long we're incubating for, which is going to be a couple days. So I'll be back a couple days from now to take those plates out. And so bacteria can grow faster than fungi, and bacteria grow better at 37 degrees Celsius. Fungi actually grow better at room temperature for the most part. Some fungi can grow well at body temperature, but the vast majority of them do not grow as well at 37 degrees, especially the environmental ones. So we can say that bacteria grow better, faster, more readily at 37 degrees Celsius when compared to fungi, when compared to most fungi. Some fungi are slower growing as well. So that's something to keep in mind when we look at our results next time we come back to lab.